Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about prokaryotic cell. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So let's talk about prokaryotic cell. So whenever we call it prokaryotic, it contains two parts. First is pro and second one is karyotic. So pro means before and karyon means nucleus. So before nucleus, that means these cells did not have any proper nucleus like eukaryotic cells. So eukaryotic cells have nucleus, these cells do not have any proper nucleus. Now bacteria and blue-green algae, these two organisms are mainly representing prokaryotic cells. Blue-green algae are also called as cyanobacteria. So mainly when we talk about prokaryotic cell, just imagine bacteria or cyanobacteria are prokaryotic cells. These cells are smaller and they multiply rapidly. That means cell division takes place rapidly compared to eukaryotic cells. And these cells are smaller than eukaryotic cells. They have variable size and shape. Like mycoplasma. Mycoplasma is the smallest bacteria. It is the smallest cell. This mycoplasma is only 0.3 micrometer in length. So it is the mycoplasma. It is 0.3 micrometer in length. Whereas there are other bacteria whose size are like 3 to 5 micrometer. So they are between 3 to 5 micrometer length. Now, if we talk about shape, then there are mainly 4 types of bacteria. Some are rod-like, those are called bacillus, like this. Some are spherical, that means they are circular type. So we call them coccus. Some are comma-shaped like these, so we call them vibrio. Some have spiral shape like this, we call them spirulum. These are main 4 types of bacteria according to shapes. Okay, so this is the structure of bacteria. Now, if you understand this picture, this only one picture, you can understand the prokaryotic cell structure very easily from only this one image. Please try to understand this image. First of all, prokaryotic cell, like bacteria, does not contain any membrane-bound organelle. Membrane-bound organelle, unlike eukaryotic cells, because we know that eukaryotic cells contain many membrane-bound organelles, like we have lysosome, we have mitochondria, plants contain chloroplast, we have endoplasmic reticulum, we have Golgi bodies, we have vacuoles, we have microbodies. Nothing is present in bacterial cell. Bacterial cell does not contain, or prokaryotic cell does not contain any membrane-bound organelles. This is the primary difference between prokaryotic cell and eukaryotic cell. Second, eukaryotic cell contains nucleus where the DNA is present. In prokaryotic cell or in bacteria, it does not have any nucleus. So this is the second most important difference between prokaryotic cell and eukaryotic cell. Now coming to this structure, we can see the prokaryotic cell or bacterial cell contains an envelope, and this envelope consists of three compartments. First one is the glycocalyx. So glycocalyx is the outermost envelope or membrane of bacteria. Glycocalyx, from the name itself you can understand, glyco means carbohydrate and calyx means coating. So glyco means carbohydrate and calyx means coating. So it is the carbohydrate coating that is present outside the cell. This is the first membrane or outermost membrane, and this is important because it helps the prokaryotic cell or bacteria to adhere to the surface. This is important for the adherence. So this is the first thing. Second, beneath glycocalyx, you will get cell wall. You will get cell wall. So cell wall, bacterial cell wall is made up of peptidoglycan. It is made up of peptidoglycan. This cell wall gives the shape to the bacteria and it also maintains the structural support. So, cell wall has mainly two importance. It gives shape and it is the structural support of bacteria. And the third is plasma membrane that is present beneath the cell wall. Plasma membrane is more or less like eukaryotic cell plasma membrane. It is selectively permeable. That means it permits some substances to enter the cell from outside and to exit the cell from inside. So it is selectively permeable, the plasma membrane. These three parts are called as envelope. Now this glycocalyx, that is the outermost membrane, it could be of two types. One is the slimy layer. Sometimes the glycocalyx is slimy. It is the loose structure, slimy structure. Sometimes it is tough and thick. That time we call the glycocalyx capsule. So if the glycocalyx is slimy, we call them slimy layer, and if the glycocalyx is tough and thick, we call them capsule. Okay, next, this structure you can see, this is the mesosome. So this is the infolding of plasma membrane and that is called mesosome. So mesosome is a very important structure in bacterial cell. First of all, mesosome helps to form cell wall. It helps DNA replication and distribution. Now, how does mesosome help in DNA replication and distribution? I am explaining it in a moment. Next, it helps in respiration because we know that bacterial cell does not contain mitochondria. Now, in eukaryotic cell, respiration or electron transport chain occurs in the mitochondrial membrane, and bacteria, they do not have mitochondria. So, where do they respire? Their electron transport chain enzymes are present in these mesosomes. In this part, respiration takes place. Next is secretion. Mesosome secretes some substances. It increases surface area because if this is the plasma membrane and if now it makes structure like this, you can see the surface area is increased. Next, it has number of enzymes. So mesosome has different roles. It has some important roles in prokaryotic cell. And this mesosome, if I zoom this mesosome structure here, we can see this mesosome has some structures like these parts are called lamellae. These parts are called lamellae, and here some vesicles are found like that in mesosome. These are vesicles, these are circular structure, and some tube-like structures also you will get in mesosome. Those are called tubules. Those are called tubules. So these are some structures we can find in mesosome. Next, bacterial cell contains cytoplasm like the eukaryotic cell. Okay. Sometimes you will get some infoldings like mesosome which are called chromatophores like this. These chromatophores contain pigments because we know that bacterial cells contain number of pigments like cyanobacteria. They require chlorophyll for their photosynthesis. Now these chlorophylls are accumulated in these chromatophores. Apart from chlorophylls, different other pigments are also there in these chromatophores in bacterial cell. Okay, now we can see this is the bacterial DNA. This is the bacterial DNA. It is naked because it is not protected inside the nucleus. It remains free in the cytoplasm. And this DNA is not wrapped in histone. Not wrapped in histone unlike eukaryotic cells. And these are circular chromosomes. So bacterial cell contains only one circular chromosome or DNA. Now we can see these are some RNA, and here you will get the ribosome. So ribosome does not contain any membrane. So ribosome is present in prokaryotic cell, but in case of bacteria you will get 70S ribosome unlike eukaryotic cell, because in eukaryotic cell we get 80S ribosome. This ribosome contains two subunits. The smaller one is 30S ribosome and the bigger one is 50S subunit. And here it is the mRNA. Now this DNA is transcribing and it is forming RNA or mRNA, and more than one ribosomes are coming to attach this RNA. That is called polyribosome or polysome. That is also very unique in prokaryotic cell, because in eukaryotic cell only one ribosome gets attached with one mRNA. But in case of bacteria, more than one ribosome are attached with the mRNA. That is called polyribosome or polysome concept. Now apart from this circular chromosome, bacteria contains some smaller double-stranded circular plasmids. These plasmids are actually non-essential parts of bacteria because they do not contain any essential gene for bacteria. In the bacterial circular chromosome, all essential genes are encoded, but in plasmids, you will not get any essential genes. Plasmids contain some extra genes like antibiotic resistance. They have some genes which take part in antibiotic resistance, but those are not essential factors, essential genes of bacteria. These plasmids, you can get one plasmid, you can get more than one plasmid, you will get multiple plasmids in a bacterial cells. These plasmids replicate in one cell and become multiplicated. But in DNA, in the circular chromosome or in this DNA, it will replicate and it will form two DNA in the next generation of cell. Like when this cell is dividing, when a bacterial cell is dividing, the DNA is replicated and each cell gets one DNA. But in case of these plasmids, they will replicate in one cell and they will become multiplied in only one cell. So that is one more difference. Next, you will get some inclusion bodies. Inclusion bodies are present in bacterial cytoplasm. These inclusion bodies are glycogen, phosphate, cyanophycean granules. So bacteria store foods in form of inclusion bodies. They store glycogen, they store phosphate, they store cyanophycean granules. And these are called inclusion bodies. And these are not membrane bound, they stay freely in the cytosol. And one more thing is absent in bacteria, centrosome. Centrosome is also a organelle that is not having any membrane and it is present in eukaryotic cell. But in bacterial cell, you will not get any centrosome. Now, one thing to mention here that I have told that this mesosome helps in DNA replication and distribution. So, how does it do so? Suppose this is one cell. Now, here is the mesosome. This is the mesosome. Now, the DNA or circular chromosome that is present in the cytoplasm gets attached with this mesosome. And the DNA is replicated. Now, the cell divides. What will happen? This cell and this cell, so two cells are formed. It also has a mesosome. This cell also has a mesosome and the DNA is distributed like this. So, the two new cells contain two circular chromosomes or DNA and mesosomes. So, in this way, mesosome helps DNA replication and especially distribution. Now, all bacterial cells are not motile. Some are motile, some are not. So, the motile bacterial cells contain flagella. This is the flagella that is formed from the cell wall. Now, this flagella contains main three parts. First one is the basal body. This basal body is attached with the cell wall. Then it has the hook-like structure and then it has the long filament and this flagella helps to move the bacteria. Apart from this flagella, some bacteria contain fimbriae and pili. So, pili or fimbriae are actually some thread-like structures like these. They are found in the surface of the bacteria. This pili is made up of pilin protein and they help the cell to attach to the host or to the surface. I forgot to mention one more thing that is very important thing, that the DNA or bacterial chromosome that is present in the cytosol, the place it is present is called nucleoid because it does not contain any nucleus. This part is called nucleoid.